Welcome to the unit on genetic and epigenetic regulation. In this unit, we will discuss the mechanisms prokaryotic and eukaryotic cells use to regulate gene expression. You'll remember from earlier in the course that not every gene in the genome is transcribed and translated at the same time. Thus, the cell must have mechanisms to regulate and control the expression of their genes. In this video, we will discuss eukaryotic chromatin remodeling. The human body contains about 200 major cell types, and although they share the same genome, they look and function differently from one another because each cell type expresses different sets of genes. Gene regulation is the process where cells control gene expression. It controls the where, when, and how much protein is produced. Each event that takes place in the expression of a gene is a potential control point for gene expression. For example, gene regulation can take place at the chromatin level through modification of chromatid or histones, at the level of transcription of the primary RNA, or its processing to messenger RNA. Or regulation can take place at the level of translation, which can involve transport of the messenger RNA out of the nucleus and controlling its stability, or translation itself. Finally, once the protein is made, there can also be regulation at the post-translational level by regulating the protein activity through various covalent modifications. If you lined up the DNA in the nucleus of one of your cells end to end, it would stretch to be 6 feet tall. This means that the DNA must be packaged in order to fit within the nucleus of each of your cells. The first level of DNA packaging takes a double-stranded DNA molecule and wraps it around a structure called a nucleosome. Nucleosomes are made out of positively charged histone proteins, which attract the negatively charged DNA. The DNA wraps around the nucleosomes like thread around a spool, forming a structure that looks like beads on a string. The nucleosomes themselves then form a helical 30 nanometer fiber, which then loops up and compresses to form the chromatin, the DNA-protein interactions which give chromosomes their structure. When DNA is packaged as chromatin, the DNA is interacting with the nucleosome histone proteins, so the transcription factors and RNA polymerase needed to transcribe genes are not able to access the promoters to start transcription. This will prevent gene expression. In order to turn on a gene, the chromatin must unravel and the nucleosomes must be moved out of the way. This can be controlled in two ways: by modifying the histone proteins themselves or by modifying the DNA through demethylation. This process is called chromatin remodeling. During chromosome remodeling, the nucleosomes are repositioned to expose different stretches of DNA. Chromatin can be remodeled by chemical modification of the histone proteins that make up the nucleosome. The portion of the histone proteins called histone tails, shown here sticking out from the nucleosome, are the areas that will be covalently modified. This occurs by the addition or removal of different chemical groups, with the two most common being methyl and acetyl groups. The most common chemical modification to DNA is the addition of a methyl group to the base cytosine. This occurs at a specific cytosine that are next to guanine bases on the DNA strand, abbreviated CpG. The P represents the phosphate between the two nucleotides. CpG sites called CpG islands are often found at regions next to a gene promoter. When the cytosines of the CpG island become methylated, transcription is inhibited. Methylation of a CpG island can change over time or in response to environmental cues, providing a way to turn genes on or off. Modifications of histones and cytosines are examples of epigenetic regulation. Epigenetic changes are changes which can affect the genetics of an organism but do not modify the sequence of the nucleotides in the DNA. These changes can be inherited but are also often reversed and responsive to changes in the environment. Epigenetic regulation is also used for silencing of not just genes, but also entire chromosomes. This is unique to female mammals because they contain two X chromosomes. For genes on the X chromosome, the dosage of genes is twice as great in females as it is in males, because males only have one X chromosome. However, the amount of protein produced from those genes must be similar between the sexes, so they must be regulated differently. This differential regulation is called dosage compensation, and it occurs through the inactivation of one X chromosome in each cell in a female, a process called X inactivation. Soon after a fertilized egg with two X chromosomes implants into the mother's uterine wall, one of the X chromosomes is selected at random and inactivated. As cell division continues, the inactivation persists, so all the daughter cells descendant from one cell will have the same X chromosome that was originally inactivated, stay inactive. As a result of X inactivation, all female mammals are mosaics. Half their cells produce gene products from one X chromosome, while the other half produces products from the other X chromosome. If the female is homozygous, there will be no difference in her cells. However, if she is heterozygous, then the cells will be producing different proteins. A good example is calico cats. The orange and black fur color is an X-linked gene with two different alleles, black and orange. So this cat's cells at fertilization would have had an X with the black allele and an X chromosome that contained the orange allele. When this heterozygous female egg replicated and went through X inactivation, some of the cells would contain the X with the black allele, but the other X would have been inactivated, while other cells would have contained the X with the orange allele, while the X chromosome with the black allele was inactivated. All cells descendant from the cell with the black allele would form black patches of fur, while all cells descended from the X with the orange allele would form orange patches of fur. The white patches seen here are from a second gene that controls patches or solid color. If the patches allele was not present, you would observe a tortoiseshell cat instead. X inactivation is why almost all calico and tortoiseshell cats are female. Many details of the process of X inactivation are still unknown, but a key player is the gene XIST. This is a non-coding gene, which means it is transcribed but not translated. The gene product is the RNA, not protein. The RNA transcribed from the gene binds to the XIC region on one of the two X chromosomes. As more and more RNA binds, the chromosome becomes coded, which triggers DNA methylation, resulting in tight nucleosome packaging. This methylation will be inherited during mitosis, so the X chromosome remains inactivated throughout the organism's life.